and new hypothesis to explain the existence of stem cells. Stem cells are unspecialized cells capable of dividing and renewing themselves and have the potential to give rise to specialized cell types, so-called differentiation. Stem cells are classified into three types. Totipotent. Each cell can develop into a new individual. These cells are found in the early embryos. Pluripotent. Cells can form any of the over 200 cell types. They found in some cells of blastocyst. Multipotent. Cells differentiated, but can form a number of other tissues. Like adult stem cells. Nearly all of the cells of a multicellular organism have exactly the same chromosomes and DNA. If every cell carries the same set of genes, how do cells become specialized in shape and function to do different jobs? This had been explained by the differences in gene expression. Only specific genes are activated and that will determine the function and the specialized structure of a cell. This transcriptional regulation is induced by special transcription factors. Fertilization occurs when a sperm and an egg have fused together to form a zygote, which begins to divide as it moves towards the uterus. The DNA in the cell is copied in preparation for cell division, and this results in two full sets of chromosomes. Each chromosome is composed of two sister chromatids. The sister chromatids are then pulled apart by the mitotic spindle, which pulls one chromatid to one pole and the other chromatid to the opposite pole. The result of DNA replication is two DNA molecules consisting of one new and one old chain of nucleotides. This is why DNA replication is described as semi-conservative. Half of the chain is part of the original DNA molecule, half is brand new. And here is my hypothesis in an attempt to explain the phenomena associated with stem cells. At the end of the process of DNA duplication, the cell has made two identical copies of all the parental DNA molecules, but in the two new daughter cells the DNA molecules are half old and half new. In a cell, the more chromosomes that contain the DNA molecule which is half old and half new, the more capability of that cell to divide and to be undifferentiated. In each round of DNA replication, each of the two strands of DNA is used as a template for the formation of a new complementary strand. Each daughter DNA double helix is composed of one conserved strand and one newly synthesized strand. The first round of replication would produce two hybrid molecules, each containing one strand from the original parent in addition to one newly synthesized strand. A subsequent round of replication would yield two hybrid molecules and two molecules that contain none of the original parent DNA. According to my hypothesis, which cell has more potential to become stem cell? In the coming slides I will make it a little bit more difficult by using two pairs of chromosomes. The probabilities of chromosomes distribution into the two daughter cells. The possibilities of chromosomes distribution into the two daughter cells in the second replication, which are 8 possibilities, generating 16 possible daughter cells. According to my hypothesis, which cell has more potential to become stem cell? Based on this hypothesis, it will be easy to explain the diminishing potentials of stem cells from toti to pluri to multipotent with the continuing cell divisions which result in reducing the extent of hybrid DNA. Also, it can be suggested that the development of any cell type is determined by the formation of a specific combination of hybrid chromosomes. And some of these cells will be saved in a nearby place to be used when needed. And with more than 70 trillion possibilities and combinations in human chromosomes, with an estimate of the total human body cells of 30 trillion, it is feasible to hypothesize that no two identical cells do exist.